Today I want to tell you about some work on barriers to infection. And so let me start by trying to set the scene a little bit. While cells and tissues and viruses are made from molecules, and we've heard a lot about the molecules involved, they also exist in the real world, and that means that they have to deal with forces and resistance and mechanical things. Say, for example, you are confronted with 3 slabs of concrete, not unlike what you see here. If you wanted to get through that or get around that, you could punch it and break it, and then you're done. So that's a barrier. You could get through it using physical means. You could also walk around it, but that's less dramatic, and so this is a more exciting way to go to deal with the barrier. My lab is largely interested in how to deal with barriers, and in fact how barriers influence the way that cells behave and operate. And so some examples of ideas that we're excited about are how force can influence the organization of actin networks. If you have a growing actin network that is generating a protrusion, pushing out a leading edge, that is also experiencing resistance, and that resistance changes the force that the network can generate, changes its density, and changes its properties. If you constrain the volume, not the mechanical force, but rather the volume that an assembling system can exist in, and this is work with Rebecca Heald's lab, the middle area here. If you simply change the volume available, you can scale the size of a spindle. And it's not because it's feeling resistance physically, but rather the limiting number of components available to assemble the spindle influences how large it can grow. And then lastly, recent work looking at how antibodies targeted to different sized molecules influence the effector function of macrophages. We find that antibody targeted to something tall is less effective at driving phagocytosis than something bound to an antigen that's short, simply because activation of FC receptors requires exclusion of a large phosphatase, CD45. So you need to create a small barrier, a small gap between the two particles in order to activate phagocytosis. So today I want to talk about the other side. Cells may have set up barriers, but viruses are very effective at getting around those barriers. And they have to do so often through clever means that allow them to exert specialized physical forces. And I'll try and explain 2 different cases where entry, replication, or release is influenced by the mechanics of the situation. And so I'll talk about 2 different viruses. One, an orthoreovirus, a non-enveloped RNA virus, and the other, influenza, an enveloped RNA virus. And this work is aimed at understanding how these 2 different viruses deal with very specific barriers. So in the case of an orthoreovirus, this is a reptilian reovirus, they want to spread from cell to cell, and they do this by driving cell fusion. And in order to generate fusion, they need to break the barrier between 2 cells. And that plasma membrane barrier is a non-trivial thing to break. If it were something easily done, we would be much more blobby than we are right now. But the integrity of that barrier needs to be overcome. And so we'll talk about how a fusion protein expressed by this reovirus is able to overcome that. And then the second virus, influenza A, must, as it comes in through the airway, overcome the barrier of the mucus in your airway and mucociliary transport that is trying to flush anything that gets caught in this mucus out of your system. And so influenza has come up with a strategy for circumventing that problem, getting through the virus, getting through the mucus that I'll describe today. And the work I'll describe is really the work of 2 fantastic students. One, Carmen Chan, who's a PhD student in the joint UCSF-UC Berkeley bioengineering program, and Mike Vahey, a fantastic postdoc who has now started his own lab at WashU in St. Louis. Okay, so I'll start actually with influenza, in part because I want to demonstrate how the pleomorphic variability in influenza could have some influence on its ability to penetrate mucus. And then we'll go to the reovirus. So penetrating mucus is not the only barrier that influenza must overcome. There are a number of different barriers, and each of these different environments require slightly different ingredients to get through. So for example, in order to get through mucus, which has much sialic acid that the virus will bind to but must get through, weak adhesion might be preferable because you don't want to get stuck in this mucus because if you're stuck, you can't move. But if you get to the target cell, you want to be strongly adhered so that you're not released from the target cell. So you have this competing desire to be strongly bound in some situations, weakly bound in others, and you've got this combination of a binding protein and a cleaving protein on the cell surface that you use to navigate those situations. So how does that happen? Well, when Mike first wanted to explore this problem, the basic challenge was in characterizing the heterogeneity, first of all. How do we know what the variability is in the amounts of HA and NA on viruses, the lengths of these viruses? And to tackle that, he developed a fluorescent strategy. Now, the normal fluorescent strategy of using genetically encoded probes is limiting in some ways because oftentimes adding a fluorophore to a viral molecule can result in poor replication ability. So it's inhibiting something about assembly of the virus. So the approach that Mike took was to use small molecule site-specific labeling that he could engineer tags into different parts of molecules to allow them to be fluorescently labeled after the fact. And then what that's allowed was him to label multiple proteins at once without having a reduction in the replicative ability of the virus. And so through a great painstaking ability to find the locations where he could insert tags and label and then produce virus, he was able to create strains where he could label 3 to 4 different proteins, quantify large amounts of virus produced from cells, and make images that look like this. So this is influenza virions that have been shed from cells, labeled in multiple colors, and you can right away get a sense of the variability in the amount of the different envelope proteins, as well as the differences in shape. And to zoom in a bit, Mike was able to label NP, HA, NA, and you can see in this case how they are quite heterogeneous in their form. Now, with viruses that you can image from large populations, you can start to characterize different heterogeneity in that virus. And so one thing he can do was look at the heterogeneity in overall amounts of HA and NA and other components, M2. Consistent with some mass spectrometry data that has been collected, there's about 5 times more HA than there is NA, and 40 times more M2. But now we have that information on a virus-by-virus basis, so we can look at the distribution of and ratio of the stoichiometries on viruses. You can also see for this particular laboratory strain that's expressing a UDORN M1, which drives more pleomorphic, more variability in shape, that you get quite a large variation in the size, the length of the virions. It doesn't change the diameter much, but the length is quite broadly variable. And what he found in a study published earlier this year was that this variability is not dependent on the number of replications. That is, each first replication you get significant heterogeneity. It's not dependent on the cell that's infected either. If you harvest viruses that are produced by a single cell, by infecting cells with very low multiplicity of infection, that variability replicates the variability of the whole population. So it seems to be on a single cell basis, you get this heterogeneous population. And in results that I won't talk about, it seems that this heterogeneity allows for more efficient release in different environments. That is, in some cases where you are better off having a low adhesive ability to a cell, your virus that has low HA is going to be better able to escape that environment. Now, with that ability to label, we're able to look at surface properties. And I want to talk about the surface properties because they affect their ability to navigate through mucus environments. Now, the first question is, how mobile is HA and NA on the virus, on the envelope of influenza? And what Mike was able to show through photobleaching experiments on a few very long virions was that there's basically no recovery. You can photobleach HA or NA, and over time, there's no exchange of the fluorescent proteins to anywhere else on the virus. So it seems like wherever that HA and NA is assembled, that's where it's going to stay for that virus. He next looked at these HA and NA labeled with this enzymatic labeling strategy. He looked at the spatial organization. And that spatial organization could be revealed by using super resolution microscopy. So rather than the turf that we'd been primarily using, he switched to super resolution to better understand what the spatial organization is of these HA and NA molecules. And what he found was that there's a bias for the NA to be on one end of the virion. It turns out that it's the end that has the nucleoprotein. But it's not evenly distributed. These two proteins are not well mixed, which is consistent with cryo-EM that has been done of individual virus particles. But now we can look at populations as a whole and see that indeed there is a bias for the hemagglutinin to be on one end and the neuraminidase to be more distributed across the other side. So what's the effect of having an enzyme that will cleave on one side and then proteins that will bind on the other side? Well, the result is motion. So in this video, that stop-start, that's not real. This is a continuous, smooth video. There we go. So what you can see is that one end of the virus, if you drop this virus down onto proteins that have sialic acid on them, the virus will scoot around in a manner that is non-random. And that non-randomness you can see if you look at any one of these movies. The red end tends to be in the rear and the blue in the front. And this is driven solely thermally. There's no energy source here. The non-equilibriumness of this is the fact that there is sialic acid on the surface. There's fuel on the surface, and at some point this will stop because it'll all be cleaved by Na as it scoots around. So when we first saw this, there was quite a bit of skepticism among us about whether this is true. So what Mike did is put together a simple model to ask if we put into a simulation the very basic ideas of the ability of HA to bind and of Na to cleave. We put in reasonable densities of sialic acid, and then we put in the key ingredient of there being thermal motion, thermally driven, and that you can't detach until you have an opportunity to step to a location where there is an attachment. So a Monte Carlo simulation replicates the ability of polarized binding and cleaving to be able to generate motion. And so with simulations like this, you can also explore variations in them. What happens if you now have a broader distribution or different arrangements? And so Mike tried out a number of different configurations for the distribution of HA and Na. You could imagine that instead of this configuration, which is kind of the idealized polarized HA and Na, what if you had a gap, a separation between where HA was and Na was? Turns out that doesn't diffuse very well. What if you had Na on both ends as opposed to just one end? That also doesn't diffuse well. What if it's in the middle as opposed to really at a pole of the virus? It diffuses a little better, but not so good. And now about punctate Na within a sea of HA, which is probably more like the viruses that we actually see. You get some bias to that motion, but not significant. And so what it comes down to, it seems, is that you need this interface between where the HA will bind and where the Na will cleave. Because if you zoom into that interface, if I have cleaved everything on the right-hand side here, the only place for my HA to be able to bind is to step forward rather than backwards, because there's no place to bind there. And so if you repeat that process over and over, you get a ratchet-like mechanism. Now, so that ability to move on 2D surfaces is something, in 2D fixed surfaces, something that we can show with fixed sialic acid. But sialic acid is not really anchored down as it is in our in vitro experiments. Really in the airway, it's diffusing within, as mucins are diffusing within mucus, there's motion of the sialic acid. On the cell surface, the sialic acid can move as well. So we next investigated with this simulation what would happen if your sialic acid were moving. Would that wash out this important separation between cleaved and non-cleaved sialic acid? And so in simulation where we vary the diffusive coefficient of sialic acid, you can see that as you might expect, if you have very, very high diffusion on the right side there, that as soon as you've cleaved a sialic acid, another one moves in. So you've not helped anything. You've not set up this gradient of binding. If you have intermediate, it actually seems to work better, perhaps because you also have new places to bind, new ways to step. So maybe the stepping forward, the binding is easier. And if you have slow diffusion, then you are more towards the fixed that we saw previously. So with variation in the diffusion coefficient, there is some optimum, and that optimum seems to be, at least from these simulations, within the realm of what you would see in a mucus gel. So this is an ability to move in a directed manner. Now, it doesn't actually tell us which direction it's going. It just says, as it begins to step, there's a bias to keep going forward. And that bias has to do with the fact that this is an elongated virion. It's not spherical. If it were spherical, it would have easier time rotating, and therefore that would remove some of the bias to step forward. So the fact that it is filamentous or more filamentous gives it essentially a rudder which keeps it going in one direction. So we next wondered whether this could have some advantage for the virus in the airway. Can you get across that mucus barrier by having your viral particles exhibit directed motion as opposed to random motion? And so what Mike did was set up a culture of airway epithelial cells, grew it at an air-water interface, grew it for a couple weeks, generated some quite attractive looking mucus. I'd never really seen mucus as an attractive material, but it's quite nice. And then what he saw with dropping viruses onto this mucus layer was that indeed they created directional paths. And so what you're looking at, this green, is marking, it's a lectin that will mark where sialic acid has been cleaved. And so you can see this path, and the path is marked out as before with particles that have neuraminidase at the one end and binding at the other end. And so if you have particles that are oriented in this way, what that can, for estimations of a mucus thickness and for reasonable densities and diffusion coefficients, the estimate is that having directed motion will allow you to cross through that mucus barrier about 1,000 times faster than if you relied on random diffusion. So by spatially organizing the molecules that bind and the molecules that cleave, the virus has this advantage in moving through something that could otherwise trap it and allow it to be cleared with mucociliary transport. So that's one example of a barrier that can be overcome by carefully organizing your envelope proteins. Now the next virus I want to tell you about is reovirus. Now this doesn't have an envelope, but it does something really interesting once it gets into cells. And that is it'll form giant syncytia. So a single virus that gets into a single cell endocytically will express a protein called P14, one of a family of fast proteins that have been discovered and described in great detail by Roy Duncan's lab at Dalhousie University. And these are pretty interesting proteins because they are so small. In the case of P14, 125 amino acids, and yet expressing that protein in a cell will allow it to fuse with its neighboring cell. So somehow this really small protein with a really small ectodomain is able to do what normally much larger proteins are able to do. So small is powerful in this case. So what Carmen Chan did was take this P14, express it into in hex cells as a model, and watch over time as these syncytia formed. And so what you can see is over the course of 12 hours and then 24 hours, you begin to see more and more multinucleated structures. If you watch this process of fusion, so if you can see, there's a membrane there. This is a non-expressing cell, and then all of a sudden it mixes with the contents mix with the expressing cell. So it's a process we can follow to some extent, and if you take a long time-lapse movie, then you generate these kind of Frankenstein hex cells that, despite the fact that they have dozens of nuclei, appear to be able to crawl around. So anyway, this is a very, very potent fusogen. So the question is how? How is it that it's able to do its work? If you compare P14 and really any of the other these fast proteins, fusion-associated small transmembrane proteins, with much better known viral fusogens or fusogens of viral origin, you'll notice that those proteins are quite tall. They're quite tall and they undergo a conformational change in order to bring two membranes close together and allow for mixing. So if P14, shown here, actual size, it's only extending its ectodomain about a nanometer or so from the surface. And it's got an N-terminal myrstillation. So it may be inserted into its own membrane, or it may be inserted into the other membrane. That we still don't really know. But what is clear is that it's really, really small. And so given that you have a forest of molecules around these molecules, which something that's overcome by these taller molecules, they can reach out, grab the other membrane, and pull down. How in the heck does this tiny little guy manage to bridge this gap and bring this ectodomain, which has some hydrophobic residues and may drive the mixing, how does it bring the two together? And so what Carmen did was first set up an assay that let her screen through various mutants. And the assay is basically expressing split YFP in cells and then infecting with P14 that if cells fuse, and the extent of fusion can be read out as the extent of YFP. So when she does that, we can measure the YFP. So over time, as you follow the infection, around 24 hours, you get significant fusion. And if you remove the cytoplasmic domain, then you get none. So the cytoplasmic domain is critical, and the same is true of the ectodomain. If you remove that, you get no fusion. So there have been suggestions from Roy Duncan that the actin cytoskeleton might be involved in this process. Now, disrupting the actin cytoskeleton can change a lot of things. And so precisely what is it about the cytoskeleton that might be involved in this process of driving fusion between a cell infected with P14 and a naive cell? So Carmen repeated some work on cytoclasin D and latrunculin, and sure enough, if you disrupt the cytoskeleton as a whole, the fusion does not proceed. Then she became a little bit more specific and looked at whiskostatin, which targets NWASP, and sure enough, that disrupted fusion. CK666, which targets ARP23, also disrupted fusion. So there's a branched actin network connection to this story, but there's not a formin connection. So a drug that targets formins doesn't seem to have an influence on the ability to fuse. So to make a longer story short, Carmen then looked in detail at the cytoplasmic tail. What is it in the cytoplasmic tail that could be linking to the actin cytoskeleton? And so using a linear motif prediction tool, she found that there was one little binding site potentially for an adapter protein called GRAB2. Now GRAB2 binds to a phosphorylated tyrosine through its SH2 domain and then has two SH3 domains that can interact with the actin cytoskeleton. So she went through and made mutants of this binding site to test whether or not Grab2 disrupting that binding could have an influence on the ability of fusion to take place. And what she found was, indeed, as you mutate, make single point mutations or a pair of mutations, you reduce the ability of cells infected with P14 to fuse with another cell. So it seems like binding of Grab2 is a critical thing. The next question is, what exactly is Grab2 doing? It has two minutes. Okay, it has two SH2 domains, and I have 2 minutes. So these two SH3 domains, one of which binds to NWASP, one of which can activate SOS. So there are two different routes to turn on the actin cytoskeleton. Through mutations, she's worked out that it really is the NWASP pathway that makes sense. And she can show this in vitro by taking the portion of the ectodomain, attaching it to a bead, mixing with cytoplasmic components, so purified components, to reconstitute branched actin network assembly, and you get an actin tail growing off this P14 tail that's dependent on the presence of Grab2. So our model of how this orthoreovirus is driving fusion is that the step that's normally undertaken by a tall fusogenic protein undergoing a conformational change to bring two membranes close together, that step is being carried out by a branched actin network that is locally nucleated from the tail of this very short fusogen. And that maybe my initial image was not too wrong, that this is requiring a very small fist that brings together these membranes, and then fusion can proceed. Okay, so with that, two different viruses solving two different problems of getting through barriers. One solving a problem of getting through a barrier, a mucus barrier, by spatially organizing itself so that it can move in a directed manner. And another virus that's getting through a cell-cell barrier to create a larger cytoplasm in which to replicate. It's doing that by not expressing a giant protein, but a very small one, and cleverly making use of the host actin cytoskeleton to drive fusion. So that sort of work is part of a broader set of projects interested in barriers and how to overcome them. We're continuing to work on understanding how the size of cell surface proteins influences downstream signaling, how the presence of proteins provides its own barrier to fusion, and with Melaniot, look how we can use diagnostics, new diagnostics to make measurements of pathogens in the field. And so I'll end with that. I've got a wonderful lab I get to work with here. Not enough space, but it's a good group. I think the funding agencies for support, and especially Carmen and Mike for their fantastic work on these projects. So I'll end there. Thank you.
Thank you for this great talk. Any question?
When does the asymmetric distribution of HANN happen? Presumably during the budding process or after release?
So it seems to be during the budding process. So because at least as soon as we measure whether there is any mobility of the surface proteins, we don't see any. So I assume that it's additional evidence for that is that if we cut off the cytoplasmic tail of Na, then we see that it is mobile at all times. So it seems to be the cytoplasmic tail in contact with M1 that's assembling, that seems to be locking in that spatial organization.
To me, it was a little counterintuitive to see that Na was at the rear end, because first when I saw the image, I was expecting that Na would be at the front end because it will be cleaving the sialic acid, and polymerization of the protein would be happening on the rear end so that it can project it forward. But it looks like that it's happening on the other way around. I was wondering if you could explain that.
Sure. Yeah, that's right. So you can imagine that cleavage would be a good way to clear out space for the particle to move. But it actually needs the sialic acid to bind. And so if, for example, I cleared out a place to step in front of me, I wouldn't be able to step there. So it needs, what it does is, if I'm clearing out the place I could step behind me, then I can't step backwards. So it's a way of preventing crossing of a path. You can't redo a step you already did. You have to go forward. And if you're elongated, so filamentous to some extent, then you're limited in your rotational ability. And so you're going to tend to move in a directed manner.
So with this image, can you tell the HA is concentrated one end and NA is other end? Because this is really totally new for me. Yeah.
Can you tell from EM?
Yeah. Can you tell EM that the HA is one end and the NA is the other end? So how? Because when you have HA and NA expressed in cell membrane while binding out, how can they tell? Selective incorporate HA to one area and the NA to the other area.
Yeah. So how does the virus do it? Yeah. I have no idea. Yeah, they're smart. There's no hope, really. We're going to get your flu vaccination, but there's no hope. No hope. I mean, it's too smart. Yeah. So yeah. Seriously though, I don't. I think that's a really important question because you can see this budding with Na focused at the tip, even when there's not an RNP there. So it doesn't seem to require the presence of the nucleoprotein. And so is there a curvature influence? Possibly. There's suggestion of lipid rafts being involved. So I think there's a number of interesting organizational influences that drive this. And there's quite a lot of heterogeneity as well.